right, last time we left off and we were talking about a plant called 4 o'clocks and how there is a gene, I don't know, we could call it the R gene, why not, where the dominant allele, capital R, causes a red flower, whereas the recessive allele causes a white flower. We're like, okay, let's take a homozygous dominant red flowering plant and a homozygous recessive, also known as a white flowering plant. 100% of the babies will be heterozygous. That means one of each allele, but that plant will not have red flowers. They will be pink because this is an example of incomplete dominance. That's where the dominant allele does not completely mask or hide expression of the recessive. So you get a blending of traits. So you get a blending of the two phenotypes. Remember that phenotype is what the trait looks like. All right, so this was to trick you all, because of course you were going to say that the flowers were all red because the dominant allele was present, and that dominates the white allele. But in this case, no, because this is a case of incomplete dominance. And I also told you to assume complete dominance. That means just normal, the way we had been doing it, unless I tell you otherwise. If I tell you something exhibits incomplete dominance on the final exam, you need to know what that means. So let's take, let's take 2 heterozygous plants now. We've done this cross before anyway, but let's do this again, where we cross 2 heterozygous. Now, heterozygous plants. So, what color flowers are each of these plants going to have? They're going to have pink flowers. Pink flowers for everyone. There we go. Yeah, so let's take a look and do this cross. We're going to need a Punnett square. That is one crappy looking square, if I do say so myself, but whatever. We're going to, pardon me. Okay, that was not my phone going off. That was someone else's phone going off. Anywho, what are we going back to here? Yes. All right, so we're doing a Punnett square. So let's put the alleles for one parent here and here, and the possible alleles for the other parent over here and over here. Let's get rid of those weird lines. And here we go. So this parent here could donate a dominant allele or a recessive allele. And this parent over here could donate either a dominant allele, we'll put it at the end of the row, or a recessive allele, we'll put it at the end of the last row. Now we're going to fill in the boxes here. Fill in these boxes by taking, like in here, we're going to put whatever is at the top of the column and at the end of the row. So we have a dominant allele at the top of the column, dominant allele at the end of the row. We've got a recessive allele at the end of the column, at the top of the column, but a dominant allele at the end of the row. And please remember you put the capital letter first. So down here we're going to have this. Down here we're going to have this. So there are several questions that, we could ask ourselves, like, what is the probability that this cross results in offspring that, let's see, are white flowering? Or maybe we could ask, what is the probability that this cross results in offspring that are heterozygous? AKA in this one, they'd be pink. And what are the probability that they are red flowering? Right there. Huh. Okay, so white flowering, white flowering. Well, these are the only white flowers right here, so it's 1 in 4. 1 in 4 chance, or 25%. Our heterozygous, well, we got this one here and this one here, so 2 out of 4, or 50%, or let's say 1 and 2. I like reducing the fractions myself. Red flowering, there's only 1 box left, so that's also a 1 in 4 chance. This is a kind of interesting thing because I don't know if anybody here gardens or has ever, hung out with anybody who likes to plant flowers, but this is the time of year that people would be planting seeds for 4 o'clocks, for example. And you can often buy a packet of seeds, and you'll get your little seed packet here, and it'll have all these picture of flowering plants on it, with their stems and leaves, whatever. But you can often buy a mix where some of the seeds will give red flowers, will create red flowering plants, some will be pink, and still others, I don't know how to draw a white flower on a white background, I'll just do it in gray. So it's a seed mix. And sometimes I wonder, do the places who create these seeds have, a bucket full of red flowering plant seeds and a bucket of white flowering plant seeds, and then a separate bucket, and they just, take the scoops out and put them into a packet of seeds like this? No, I've read that a lot of places will just set up a cross between 2 heterozygous plants here and here, and then they'll just put the whole mix, they'll harvest all the seeds and then just put it in there into one packet. And I have noticed that when I buy certain packets of seeds, one color will dominate, like there'll be more pink than anything else, and that kind of makes sense if they did it this way. So kind of fun to think about that, nursery companies and seed producing companies can use this kind of cross to their advantage to make life a little easier, so that they can just scoop their seeds out of one mix bucket instead. Be kind of cool. All right, so I showed you an example of incomplete dominance to show you that things are not always straight up super duper easy. There's a dominant allele and a recessive allele, that sort of thing. There's often more that goes on. So a few examples of things where it's not quite this simple is incomplete dominance. So that's a little unusual right there. So, things are not always super duper simple. Let's write that down. Inheritance is not always, your basic, what we call Mendelian inheritance. One dominant allele, one recessive, etc., yada, yada, yada. All right, so examples that we've had of that are, the examples of this are incomplete dominance. That's one example. All right. Another example. Let me number these. There's one. Incomplete dominance. 2. For example, there is another type of inheritance where it just it's way more complicated than, tall peas, short peas, that sort of thing. And that would be for an example of human eye color. All right, human eye color is not always the easiest thing to predict. And that is because eye color is controlled by more than one gene. So we call this a multigenic or polygenic trait. And with multigenic and polygenic traits, more than one gene each with its own set of alleles controls a trait. So this is when a trait is controlled by more than one gene, each with its own alleles. So for example, human eye color. It's controlled by 3 genes. Yeah, 3 genes. At least that's what all those eye color calculators that I find online. You can find a human eye color predictor online, and you put in all of your data and your parents' data and their siblings' data, and you can see like what the chances were of you having brown eyes or green eyes or blue eyes. Yeah, I think hazel eyes count as green eyes in these calculators. But it's really very interesting. So it's controlled by 3 genes, like let's say the A gene and the B gene, and let's say the C gene. I'm just making up this A and B, but like there would be 2 alleles, a dominant and a recessive allele at the A gene, a dominant and recessive allele at the B gene, a dominant and recessive at the C, that sort of thing. And the combination of these 3 gives you different eye colors. So it's very interesting. So eye color is an example of that. Skin tone, skin color is a total example of that as well. Skin color is controlled by, I believe it's 6 sets of genes, each with their own allele. So it's very difficult to predict skin color often, especially with people who are multiracial. For example, like a very, very pale-skinned person having children with a very, very dark-skinned person, all of their children would be somewhere in between, would have a medium skin tone. But when 2 people of medium skin tone have children together, they can have children with a skin tone ranging from very light to very dark. So it's very complex. This is a multigenic, polygenic trait. Another example of this, and this is a lab that you would have had, is coat color. Oops, I'm getting ahead of myself here. Coat color in cats. In other words, the color of cat's fur is determined by so many different genes. I think it's like controlled by 10 genes. Controlled by about 10 genes or so. It is insane. And that's a really great lab, and I'm so bummed you all didn't get a chance to do it, because it's a really fun lab too, and we get to look at all kinds of cat colors. But hey, that's the way the pandemic rolls. But I do want you to know that coat color in cats is an example of a multigenic or polygenic trait. Skin color, human eye color. So none of these are easy to predict. It's a very complex inheritance pattern. All right, so things are not always that easy. Because sometimes things exhibit incomplete dominance, some traits are controlled by more than one gene, and sometimes you have something called co-dominance. Co-dominance is kind of similar to incomplete dominance in that you can get a blending of traits, but co-dominance goes a little further. It's not that one is recessive. There are no dominant and recessive alleles at all. So no even partly dominant alleles. No partly dominant or recessive alleles here. And I need to switch the color for this because the example of this is human blood types. Human blood types exhibit co-dominance. So we gotta talk about human blood types and how they work. So you probably know, or you may have heard, that that you can't just receive blood. Let's say you need to receive blood. You're in the hospital and you've lost blood for some reason and you need to receive blood. You cannot receive just any blood. Pardon me. Okay, apologies for that once again. That was another person's device. I had motioned to them to turn it off and they apparently did not do that. But no issues here. Ha ha ha. Human blood types. Okay, you cannot receive blood from just anybody. You need to receive blood from someone who has the same blood type as you or someone who is O negative. Okay, and that's because type O blood is what we call the universal donor. I don't know if any of you have ever donated blood, but if you are type O blood, you're probably really popular with the Red Cross. They probably email you and call you saying, oh, we need your type O blood. Please come and donate, because anyone can receive type O blood. If someone with one type of blood receives the wrong type, you can get really, really sick. And that's because you have an immune reaction against receiving the wrong blood type. So human blood types have to do with your immune system. So you have to be really careful about this. So there are different types of blood. So there is type A blood, type B blood. Anybody here type AB blood? And I bet there are a bunch of people here who are type O. Alrighty. So let's talk a little bit about this in a different color. Type O blood is what we call the universal donor. They can donate blood to type AB, to type B, to type A, and to type O people. Everybody can receive type AB. I'm sorry, everybody can receive type O blood. Right. Whereas type AB we call the universal recipient. People with type AB blood can receive blood from type A people, from type B people, from type AB people, from type O people. There we go. And here's why. Because this has to do with immunology. No, forget that. Let me go back. Let's talk about what makes type A blood, and what makes type B blood. And to show you this, people with type A blood have red blood cells. And I'll kind of draw it like a little oval this way. You ever see them drawn? They look like they have a little dent in the middle, maybe. They're kind of disc-like. If these are your red blood cells here. And I'll draw red blood cells for each of the types here. Okay. There we go. People with type A blood have proteins on the surface of their red blood cells that are what we call type A proteins. And so I'll just draw them as these little spiky things, like that. These are the type A proteins, these little spiky things, A proteins. Type A blood proteins. And they're on the surface of your cells. Sometimes they're called surface antigens. Now, people with type B blood, they don't have these little type A spiky looking proteins. They have something else. They have a B protein, and I'll just draw that B protein like a little blue ball. Yeah. So that's our type B protein right there. people with type AB blood have both the type B protein on the surface of their red blood cells, but they also have the A protein. Like that. So they have both. So the thing is, these proteins that are on the surface of these red blood cells are recognized by your immune system. And let's say you have type AB blood. So your red blood cells look like this one here. All right? So now that you're looking at that one, I'll erase those arrows. Let's say someone gives you blood from a type A person, gives that to you. The type A blood has these little pink spiky A proteins on there, but your immune system is cool with it because your immune system already is comfortable and familiar with having these spiky A proteins. So you're good. People with type AB blood have no problem receiving blood from a type A person because their system, your system, is used to seeing these A proteins. Likewise, if you give an AB person blood from a type B person, your cells are totally going to be cool with it because your cells already are familiar with these B proteins that are all over the B blood cells. But think about it for a second about what would happen if you gave type B blood to a type A person. The type B blood has these little blue B proteins on them. But the type A person doesn't have those blue B proteins on them. Their immune system is not familiar with them. So this person with the type A blood, their immune system is going to freak out going, what the heck are these blue proteins on there? Attack it, get rid of it, and you're going to have a huge immune response and get really, really sick because your immune system is freaking out that it doesn't know these B proteins at all. All right, so clean this up a little bit. Here's the deal with type O blood. Type O blood, notice I didn't draw any type O blood down here. Down here, type O blood, this is what we're looking at right here. Right there. People with type O blood don't have O proteins. There is no such thing as an O protein. There is no O blood protein. Type O blood is just the absence of A proteins and the absence of B proteins. So type O blood has no surface proteins. Well, they might have others, but they don't have they have no A and no B proteins at all. It's just the lack. It's the lack of A or B. Let's even say it that way. That's better. Say it this way. Lack of A or B proteins. So can you see that if someone gives type O blood to a type A person, the type A person's immune system is going to be fine with it because there's nothing funky. There are no weird looking proteins that it's never seen before on the surface of the type O blood. Likewise, type O can donate to type B because it just doesn't have anything that's going to make the type B person's immune system freak out. And likewise, no problem giving type O blood to type AB people because the type O blood doesn't have any funky looking surface proteins on it that the type AB person's immune system has never seen before. So we call type O blood the universal donor because they can donate to everybody. But think about it for a second. What type of blood can a type O person receive? Can a type O person receive blood from a type A person? No. No way, because type A blood's got these spiky looking proteins on them, right? And the type O person's immune system is going to be like, what the heck is this spiky looking protein up here? Freak out, everybody, attack, attack, attack. You have this huge immune response and you get really, 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 really sick. So you can't do that. Likewise, you cannot give a type O person type B blood because their immune system has never seen these little B proteins. And likewise, nope, do not ever, ever, ever give type AB blood to a type O person because their system has not seen the B proteins or these spiky A proteins at all. Okay, so these are immunoproteins, really. They're involved in the immune response. So we need a way to write these alleles. So the way we write these alleles, and I'm going to scroll down just for a little bit. I'll scroll back up in a moment. We're going to write the letter capital I for immune system. And for the A allele, we're going to put a superscript capital A right there. We are not going to write things like A and B and O. There is no O anyway. It does not work that way. And the thing is, there is no dominant A allele and recessive A allele. Nope, does not work that way. This is codominance, and there is no clear-cut dominant and recessive allele at all. So we write a capital I with a superscript A for the A allele, and this causes someone to have type A blood. For type B blood, for the B allele, we're going to do the same thing and write a capital I with a superscript capital B, and that causes type B blood. Now here's the deal with the O thing. There is no O allele. The O type O blood are just people that don't have an A allele or a B allele. They just have nothing. There is no O, I with a capital O. That we don't write it that way. It's because this implies that there is something there. There's not anything there. So we're going to get rid of that right now. So how do we draw the lack of A or the lack of B? The way your book does it, and the way most biology books do it, is to write a lowercase scripted I, to use it as a placeholder. This means there is no A allele and there is no B allele there. So if you see a lowercase script I, it's a type O. Now remember way back to Mendel. And we're talking about Gregor Mendel's stuff and the whole thing, every individual has two factors, has two alleles per trait. So every person is going to have a combination of 2 out of these 3 options. So let's scroll back up here to, it looks like we're pretty much just having a table here, aren't we? This is what the blood types look like in this column here. These human blood types, these are the phenotypes. Let's put the genotypes that are possible for each type in this column. So, type A. A type A person is going to have 2 out of 3 of these, right? So, a type A person could have I with a capital A. And their second allele, one that they inherited from their birth mother, one that they inherited from their birth father, would be like that. But are there any other combinations of these 3 that will lead to type A blood? Could a person, for example, have this and one of those and be type A? No, doesn't work. We already determined you can have 2 of these, but what about one of these here and one of these? And the answer is yes. Type A person could be capital I, superscript A, I, or they could be I, A with a scripted I. Like that. So that's a possibility. Likewise, type B. Person could have two B alleles. Or a type B person, and we're talking, I'm sorry, I shouldn't be saying maybe type A person and type B people. I'm not talking about, people who are, type A in psychology and type B in psychology. We're talking type A blood, type B blood. But a person with type B blood could have one B allele and then one non-allele. All right, so we put the little script I. Okay, type A, B people, there is only one option, isn't there? People with type AB blood have to have one A allele and one B allele. There are no other options. If we replace this one with an A, they're going to have type A blood. If we replace this with a lowercase I, they're still going to have type A blood. So, type AB people only have one option. Likewise, type O folks only have one option either. They have to have two non-alleles. That's it. Yeah. So, before the pandemic, what I normally do is tell people, you got to memorize this. If you can memorize this table here, you're great for doing all of these blood type problems because everything's in there. Well, make sure you took notes on this and wrote it down because we're going to need to use this info here to do some problems. So let's see. Let's do some problems here. So let's scroll up here and let's say two people with type AB blood have kids. What is the probability they will have a child with, let's say, type AB blood? All right, so if the people are type A, both type A, scroll back here and take a look. This is, type AB. This is the only possible genotype for each of them. So what we're doing, really, is crossing someone with type AB blood with someone with type AB blood. So we're going to do a Punnett square. Oh, it's going to go right through the middle here, this midline. I'll just use this line anyhow. So what we'll do is we'll look at this parent over here and say they can donate this allele to their kids. We'll put that A allele right there. Or they could donate this B allele to their kids. This parent over here, they can donate either an A allele or a B allele. And we'll put that B allele at the end of the column there. Now we're going to fill them in. Okay. Here we go here. Right there. This kid's going to have AB. Yep, and this kid, yep. Something like that. So wait, what did we ask here? They will. What is the probability they will have a child with type AB blood? This person's got AB blood. This square's got AB blood. That's 2 out of 4, or 1 in 2, or a 50% chance. Notice also they have a 1 in 4 chance of having a kid with type B blood and a 1 in 4 chance of having a kid with type A blood. Is it possible for this couple to have a kid with type O? And the answer is no, they can't have a kid with type O. That's just not how it works unless you have some weird germline mutation with both parents. Not happening. There we go. So that's an interesting cross. Let's look at another one. Let's let's say, let me ask you something else. Can two people with type B blood have a kid with type O blood? Can they? Is it possible? Let's scroll back to our chart and look. We don't even need to set up a Punnett square for this, do we? Because what are the options, okay? Is it possible? Now, we don't know if these people are this, which would be having two B alleles, or if one person is that and one person is this here. I'm just asking you if it's possible. And the answer is yes. If both people with type B blood have one B allele and one non-allele, then yes, of course they could. So if we crossed two people who are both that, well then yes, it's possible. And I'm not saying that every couple who both have type B blood are going to have one B allele. Put that one here, put this one here. Same. But of course it's possible. There's a 1 in 4 chance of type O right here. Right there. Can this couple have a kid with type A blood? The answer is no. No, because where'd that A allele come from? This person doesn't have an A allele to give to their kids. This person doesn't have an A allele to give to their kids. So the answer to that is nope. The answer to this one for type O blood is yes. Yes, they can. There's a 1 in 4 chance of that. So yeah, one more. Let's see. Let's just do one more. Let's say a woman, and I'll use the universal symbol for woman. A woman with type B blood has a kid with type O, and we already know that's possible, right? Let's say she tells a man with type AB blood, it's your baby. Is this even possible? And the answer is no. What I'm asking you is, can a person with type AB blood have a kid with type O blood? And we've determined no. Because this person with type AB blood, their genotype is this right here. They have one A allele and one B allele. They can either give their kid an A allele or a B allele. But this kid is type O. Type O is this right here. They don't have an A allele or a B allele. So the answer is no, he cannot be the father. Back in the days before we had DNA sequencing and DNA fingerprinting, we would have to do paternity tests by looking at blood type. So that was one way that we would look into it, or you might even look at blood type first before you do expensive DNA testing. So on the final exam, you will have a couple of blood type problems to do, so just make sure you know how this works and be able to do crosses based on this table that we created right here. And please know that this is an example of codominance. Codominance. All right. Another example of when things get funky, when they are not super straightforward, with one dominant allele and one recessive allele, we're going to look at a concept called epistasis. Epistasis. Epistasis is a phenomenon that occurs when the alleles at one gene influence the expression of the alleles at a whole other gene. Or let's say when the alleles at one gene, let's say overrule, that's a better word for it, overrule the alleles at a whole different gene. Yeah. So the best way to show you this is to give you an example. So let's go with this. And let's think back to coat color in cats. Oh, so alliterative. Coat color in cats. Coat color in cats is a multigenic trait. We already talked about that a little earlier. And it's controlled, meaning it's controlled by many genes. And one of the ways it's controlled by many genes is through this epistasis thing. So let me show you an example of that. So for example, there's a gene called the B locus. Ooh, new word here, locus. Locus means the location of a gene on a chromosome. Yeah, the B locus. The B gene, the B locus. B locus is more is the more accurate term. So locus and location have the same root word, so that's where that comes from. So the B location, the B locus. There is a dominant allele, capital B allele, that causes a cat's fur to be black. So causes black fur. There we go. Whereas there is a recessive allele at the B locus that causes a cat to have brown fur. So I don't know if you've looked at too many cats, but it is more common to find a black cat than a brown cat. Usually there are tons of black cats all around the world. So for example, if a cat is homozygous dominant, what coat color is that cat going to be? Its fur is going to be black. Got that? If the cat is heterozygous, and we're assuming complete dominance because I didn't tell you it was incomplete dominance, so this cat would also have black fur, have another black cat. So the only way to get a cat with brown fur is for it to be homozygous recessive. So that'll give brown fur on a cat. So fine, that's great. There is another gene locus in cats called let's use a different color. It's the C locus. And the dominant allele, capital C there, allows a cat to have its normal coloring. Normal pigmentation, let's call it. Whereas the recessive allele, and I'm going to write a little lowercase c and put a little like hat on it like that so that we can tell the difference. This causes a cat to be an albino. An albino cat has all white fur. All white fur, no pigmentation. And a cat that is a true albino even has no pigmentation in their eyes. So this type of albino, there are other types of albinos in cats, but this particular type of albino would actually have red eyes. They would not have any pigment to the iris in their eyes, so the red blood vessels would show through. So those cats have red eyes. Yeah, I know, it sounds kind of freaky, but whatever. If you're into cats, you're fine with red-eyed cats, I guess. Kind of freaks me out a little bit. All right, so whatever. If a cat has two dominant alleles, it has normal pigment, right? If a cat is heterozygous at the C locus, they will still have normal pigment. But if a cat has two recessive C alleles, they will be an albino. So let's put together the B locus and the C locus. All right, let's say we have a cat that is let's make it homozygous recessive at the B locus and homozygous at the C locus. What color fur is this cat going to have? Well, the B locus says it's going to have brown fur, and the C locus says it'll have normal pigmentation, so this cat will have brown fur. Easy peasy. Well, let's say another cat is homozygous recessive at the B locus, but let's say at the C locus, it's also homozygous recessive there. Now the B locus here says it's going to have brown fur, but the C locus says, not so fast, you're going to be an albino cat, and I don't care what the B locus says. That's because the C locus is epistatic over the B. Epistatic. C locus rules. The C locus has the final say, or it has more of a say in what color that cat's fur is going to be than the B locus. So the C locus is epistatic over the B locus. Doesn't matter what the B locus says. If the B locus says it's going to be a brown cat, but the C locus says, not so fast, you're going to be an albino cat, that cat's going to be an albino, period. That is epistasis. And with coat coloring cats, there are like 10 more genes than this that help determine whether a cat, has a tuxedo cat with a white tummy and white tips of the paws, or if it's a calico cat, or if it's a tabby cat, or if it's a tabby cat with a white tummy, or if it's a brown cat, or if it's a Siamese cat. Oh my gosh, it's crazy. So, but this is just basic epistasis that I would like you to be aware of. Yeah. So right there. Okay. So let's see. We are going to move on a little bit. We have a little clean up work to do. What I want to talk about next is okay, it's nice to talk about coat coloring cats. It's nice to talk about 4 o'clocks, human blood types. These aren't things that we tend to think too much about, or things being critical to our health. Like, a cat's health is not affected whether or not it has brown fur or black fur or is an albino or a tabby cat, whatever. Our health is affected by our blood type, because we want to know what type of blood we can receive. But for example, when a woman is pregnant and people ask, are you hoping for a boy or a girl? The answer is always, it doesn't matter. I just want a healthy baby, right? I know that sounds very cliche, like I don't care, I just want a healthy baby, because because you just want a healthy baby. And because there are a lot of human genetic disorders. There are lots of inherited traits that can affect human health. So we're going to look at some human genetic disorders. And the inheritance pattern is going to be a little different. All right, so to give you a little bit of a background here, because we did not have time to do mitosis and meiosis in the cell cycle. That is not going to be on the final exam. I know it's in your syllabus, but we had to skip over that because I cannot expect you to learn twice the amount of information in half the amount of time. So we had to skip that. So I got to catch you up a little bit on vocabulary. All right, so what we need to talk about is I need to give you a little bit of a background for human genetic disorders. We need to talk about chromosomes. Okay, your chromosomes are in your nucleus of each and every one of your cells. And a human cell Yeah, let's stick with orange. A human cell. I'm going to draw the nucleus ridiculously big. Has 2 of each chromosome. And they come in pairs. And I'm drawing chromosomes like these squiggly little lines. There, this squiggly little line here means chromosome. It's my fabulous artwork, yet again. And we have a bunch of pairs of these chromosomes. Does anybody know how many pairs of chromosomes humans have? We have 23 pairs. Humans have 23 pairs, right, for a total of 46. Well, there's not enough room in this nucleus here for me to draw all 23 pairs. Like, we're running out of room there. Okay, but we number them. We give them a number. Like, there's human chromosome 1, and we have a pair of human chromosome 1s. Where we got this one from our birth mother, and one from our birth father, right? So this is from your birth mother, and the other one is from your birth father. Universal symbol for female, universal symbol for male as well. We also have a pair of chromosome number 2. Fabulously exciting names that scientists gave all of the human chromosomes, but it's just easier to take care of them than if we give them all a number instead of calling them, I don't know, Kim and Kylie chromosomes. So let's see, we got a pair of 3s, we have a pair of 4s, all the way up to, because I can't draw all of them in here, all the way up to the very last pair of chromosomes, which do not get a number, right? What are they? Remember? They are the X and the Y. Remember? So I'm going to put an X for this one and a Y here. Oops, I didn't draw them quite correctly. There we go. There we are. Ha. Notice that I drew them a little differently, and that is because the X chromosome is much larger than the Y. There are way more genes on an X chromosome than a Y chromosome. But we number all of the chromosomes, and we got, this chromosome from our birth mother, this one from our birth father. We got this chromosome 4 from our birth mother, this one from our birth father, etc., etc., etc. Okay, so this cell here is male, right? Its sex is male. Who did this male inherit their X chromosome from? From their mother, because males don't inherit their Y chromosome from their birth mother, right? Females have 2 X chromosomes. Males have an X and a Y. So it's up to which sex chromosome is in the sperm, what the sex of the baby is going to be. So you had to have inherited from birth dad right there. Why am I giving you all this information? Because we need to have a little more vocabulary here. And I just want to show you, there is a word that we use to refer to the X and the Y chromosome. So the X and the Y, these are sex chromosomes. And we call them that. But all numbered chromosomes, not hashtagged chromosomes, all numbered chromosomes are what we call autosomes. These are called autosomes. It sounds like AutoZone, a place where you take to get your car's oil changed, but no, this is autosomes. Autosomes refer to chromosomes that are numbered, chromosomes that are not sex chromosomes. And human genetic disorders are inherited differently depending on whether or not the gene that we're talking about is on an autosome or on a sex chromosome. All right, so that's the background for human genetic disorders. That gave us a little bit more vocabulary there. There are different types of human genetic disorders. All right, so what we call genetic disorders are caused by what we call a faulty allele. And the inheritance pattern is going to be different if the faulty allele is on an autosome or on a sex chromosome. So we got different types of human genetic disorders. There are, for example, let's switch up. What haven't we done in a while? Let's go with this reddish color here. There are, so types are what we call autosomal recessive disorders. So this is where the faulty allele is recessive and on an autosome. So an example of this, an example of an autosomal recessive disorder is something called cystic fibrosis. Cystic fibrosis is a lung disorder, and it is characterized by people who, it is characterized by folks who get a really thick mucousy, a very thick salty mucus in their lungs, and it's because chloride ions are not allowed in and out, are not pumped in and out of the lungs effectively. So they end up with a lot of lung problems. 40 years ago, having cystic fibrosis was pretty much a death sentence. Most people with cystic fibrosis did not live past adolescence much at all. But medication has improved so drastically, and also there has been some genetic therapies that have been very, very promising and helped people with cystic fibrosis. So people with cystic fibrosis nowadays are expected to live, live a much longer lifespan than they used to, well into adulthood and beyond, if not, into their 80s now. But the dominant allele, I'll give it an F for fibrosis, causes a normal phenotype, but it's the recessive allele that causes fibrosis. So there are 3 genotypes, are there not? There is homozygous dominant, there is heterozygous, and there is homozygous recessive. Which one of these genotypes results in fibrosis? This one here, fibrosis. So the affected individual. Let me write this a little differently. Affected individual. They have the disorder. Has disorder. Is always homozygous recessive for autosomal recessive disorders. All right, so, but this person over here has got a normal phenotype. This one here's got a normal phenotype, but there's something special about this person who is heterozygous. And here's where someone in class will usually say, oh, they're a carrier. Yes, they are a carrier of the disorder. For someone to be a carrier of something, it means that they carry the allele for it, but they don't have the disease. Carry the allele, but don't have the disorder. Right? They're a carrier. Now, if someone has cystic fibrosis, what do we all automatically know about their parents? If someone has the disorder, what do we know about their parents? Where did they inherit this allele? They had to get it from one parent, let's say maybe mom. And they had to get this allele from the other parent, so dad. So that means dad had a recessive allele and mom had a recessive allele. But what if the parents are like, don't have the disorder? They're like, what are you talking about? How could we have a kid with cystic fibrosis? Neither of us has it. That means that they must have been carriers, huh? It's a possibility. So let's look at this. Let's say, let's switch colors. Okay, let's say, a couple has 2 children. Their oldest is of normal phenotype. So, meaning normal, they don't have the disorder. Their second child was diagnosed with cystic fibrosis. What's everybody's phenotype here? Or sorry, what's everybody's genotype here? Well, we already determined, okay, that their second child is diagnosed with cystic fibrosis. That means their second child must be homozygous recessive, huh? Okay, one more thing we need to know about the parents. We do need a little bit more information here, don't we? A couple, each of normal phenotype, has children, 2 children. Their oldest has a normal phenotype, but their second child was diagnosed with cystic fibrosis. Okay, we know back here, those parents had to have been heterozygous then, right? So the parents must be heterozygous. So when they had kids, let's do our Punnett square here. Come on up. This parents' alleles go here. They could donate a dominant allele to their children or a recessive. This one could donate a dominant allele or a recessive. Fill in those boxes. What are the chances that they would have a kid with cystic fibrosis? It's 1 in 4. And, you know, you're the genetic counselor explaining this to their parents, trying to explain to a set of parents why their child has a very difficult, challenging disease. And you explain to them, but we don't have the disorder. And you're like, well, but you each are a carrier of the faulty allele right there. It's just that each of you have a dominant allele that keeps you healthy. They're like, but our oldest child doesn't have the disease. And you're like, well, what's your oldest child? They're like, well, there was a 3 in 4 chance that they'd have a normal developing child. There we go. But so we know that, and they're like, well, but what about our oldest child who's normally developing? Are they heterozygous or are they homozygous dominant? And the answer is, well, we don't know. We don't know by looking at you. We'd have to do some genotyping there. We'd have to do some DNA sequencing where you would send off your stuff to, 23andMe, and they would send you back all of your data. But just looking at the chances, what are the chances the oldest kid is a carrier? What are the chances that the oldest kid is a carrier? And if you said, well, here's a carrier and here's a carrier, that's 2 boxes out of 4, so that's 1 into a 50-50 shot, the answer to that is no. Because here's the deal with that. We can rule one of these boxes out, can't we? For example, we know this person is not homozygous recessive because they don't have the disorder. So we can just get rid of that, right? This box doesn't exist for this person anymore. So really, it is 2 out of 3. This person is twice as likely to be a carrier than not. So it's actually a 2 in 3 there. So very interesting. Interestingly enough, another example of an autosomal recessive disorder is sickle cell anemia. Sickle cell anemia. There is a gene that causes your red blood cells to have a normal shape, and that's a dominant allele. So normal red blood cells, let's write RBC for red blood cells, whereas the recessive allele causes a person to have sickled red blood cells. And if you like the pictures that I drew of red blood cells before, normal ones look kind of like a disc like that. A sickle means like a little crescent shape. And people with sickled red blood cells don't carry oxygen very well. So people with sickle cell anemia, they don't get all the oxygen that their cells need, which means they don't make all the ATP through chemiosmosis that their cells really, really need. So it's a problem. But can you think of what demographic in the world is more affected by sickle cell anemia? There is one. Yeah. It's people of African heritage. African Americans and people of African heritage tend to have, and it's only because the recessive allele is more prevalent in certain parts of the African continent historically. And here's why. You can have a homozygous dominant, normal phenotype. You can have a heterozygous. So this would be normal, but they're a carrier, right? Carrier. And then there is the homozygous recessive, and you have the disorder. You are affected by the disorder. So you have sickle cell anemia there. Okay, back in the day, we have treatments that are possible for people now with sickle cell anemia, but, hundreds of thousands of years ago, when we didn't have modern medicine, people who had sickle cell anemia generally wouldn't make it to adulthood and would not be able to pass on their genes to offspring. So you would think that this faulty allele would die out, but it didn't, because in certain parts of Africa, malaria is very prevalent. And there is something about being a carrier, there's something about being heterozygous that also gave people some immunity to malaria. So this also protected against getting malaria. And people used to die of malaria. It's a serious disease, serious infection. So it was very advantageous, before the dawn of modern medicine, to be heterozygous. And so this recessive allele stuck around in the population right there. So, yeah. And there's also, by the way, this does show, it does show some incomplete dominance. It does display some incomplete dominance where people who are heterozygous, if you're homozygous recessive, your blood cells would be all sickled shape. Normal over here, there'd be normal shape. You might have some sickling to this one. And so people who are heterozygous might not even know that they have even the slightest bit of sickling to their red blood cells unless they went to, say, a very high altitude place where there is a little less oxygen available in the air to breathe in. And if you're exerting yourself there, like, say, a football player playing in Denver, playing against the Denver Broncos. Exerting yourself at a high altitude when your cells have some slight sickling to it could mean that your cells are not getting the oxygen that they need. Fermentation's not going to help you out for very long. And there was a football player, gosh, I think it was like 20 years ago, playing for the Pittsburgh Steelers. I can't remember the guy's name, but they went to play in Denver, playing against the Broncos. And I guess it was Mile High Stadium at that time. But the guy didn't know that he was a carrier for sickle cell anemia, and he collapsed. He actually had to have his spleen removed because it did not get the oxygen that he needed. So, I mean, this is really serious stuff. This guy lost a body part. Thankfully, it wasn't one that he needed that desperately. But after that, his physician benched him, saying, "You are not allowed to ever play at a high altitude like this again. It's life-threatening for him." So that's some incomplete dominance, as well as being an autosomal recessive disorder, as well as being a carrier. So there is that right there. Now we will pick up on Thursday and talk about autosomal dominant disorders. So I hope you have a great day, and we'll be back. And don't worry, don't be emailing me with questions about the final. I will post stuff about it. I'm just trying to get my act together with it and trying to figure out how far we're going to get tomorrow. But your final exam will be available for you during the final exam period, and I will probably give you a 2-day window for it, not just the 1-day window. I don't know. I'll be a little more flexible there. I'll give you a 2-day window. I just don't know when that window is exactly, but I will let you know on Thursday. So be safe, stay home, wear masks. You know that Governor Baker has insisted on people wearing masks if they're going to be in public and not able to socially distance. So make sure you get yourself some masks. If you don't have one, use a bandana. Don't use a t-shirt. They don't work as well. Use a tightly woven fabric, fold it over, tie it around your face if that's what you got. Anyhow, there was your public service announcement. I hope you have a great day. Bye-bye.